So you know how we breathe oxygen? Well, it was once toxic to almost everything on Earth. So let's talk about our journey from hating to loving oxygen. The first organisms on Earth were potentially chemoheterotrophs that used abiotically produced organic carbon to do their metabolism, or chemoautotrophs that used inorganic carbon and were able to convert it to organic carbon. In any case, they used some sort of chemicals, that's why they're chemo, auto, or heterotrophs, to do their metabolism. They took up some sort of, chemical, and in combination with one of these carbon sources, were able to gain energy from a metabolic process that used these compounds. So what were these chemicals? Well, early life probably reduced chemicals such as either sulfate, elemental sulfur, oxidized iron, carbon dioxide, or nitrate. And as primitive chemoautotrophs died, like those that reduced these compounds by using hydrogen as an electron donor, these guys would have provided organic carbon to chemoheterotrophs by their carcasses. And that was the birth of fermentation, which allowed microbes to expand to areas surrounding hydrothermal vents, assuming a hydrothermal vent life origin, which is a big assumption. So take this with a grain of salt, but assuming that they were able to expand to wherever the organic carbon was being dispersed. And you might think that this sounds repetitive because you might have seen my first life video or earliest life video where I talk about all of this. So if you want to know more about all of this stuff with more details and how this first life came about and what it might have been, you can see that video. But moving on now to more of what types of organisms were around when oxygen began to increase and how these anaerobic and not oxygen using and not oxygen liking organisms coped with that. So methanogens, which live today and produce methane from CO2, were likely around on early Earth, as were SRB, or sulfate-reducing bacteria, that reduce sulfate to produce hydrogen sulfide. These organisms early on likely used hydrogen-2 or molecular hydrogen as their electron donor to reduce these compounds. If you don't know, chemically reducing compounds just means giving those compounds electrons, and chemically oxidizing compounds means taking away their electrons. So to reduce the sulfate to sulfide and to reduce the carbon dioxide to methane, they needed to give those compounds electrons, and they needed what's called an electron donor, like hydrogen, to do that. And because they both needed this electron donor, they were competing on early Earth for this substrate. However, it wasn't until oxygen rose that methanotrophy, the complementary process to methanogenesis, in that instead of producing methane by reducing it, they oxidize the methane and produce carbon dioxide. And then also, because of oxygen rise in the atmosphere, there was an increase in oxidative weathering of continental material that transported more sulfate to the oceans and led to a greater amount of sulfate reduction by SRB. And therefore, methanogens that had outcompeted SRB early on, when there wasn't too much sulfate transport to the oceans, started to become of lesser importance, and sulfate-reducing bacteria started to outcompete methanogens and become more important. That marked a switch in atmospheric conditions to less methane, more carbon dioxide, and in oceanic conditions, it marked a shift to more sulfidic, or what's called euxinic conditions, because things were still relatively anoxic or oxygen depleted, where SRB were active in creating sulfide, and they also really just don't like oxygen, so they lived where oxygen wasn't, and oxygen at the point was still, mostly the surface waters, and so these SRB made the bottom waters euxinic, or just anoxic and very sulfidic. The invention of methanotrophy also had major implications for nitrogen fixation. So let's talk a little bit about the nitrogen cycle and how this worked. Nitrogen 2, which is abundant in the atmosphere and has been for all of time on Earth, reacted with methane to form hydrogen cyanide, which then in solution formed ammonium. Ammonium was then oxidized to nitrite and then nitrate by ammonium oxidizing and nitrite oxidizing bacteria. And this conversion of ammonium, or loss of ammonium, because it's becoming converted, and the decrease in methane due to methanotrophy evolution necessitated nitrogen fixation, or the ability of organisms to fix nitrogen directly from the atmosphere as molecular nitrogen 2 into bioavailable organic nitrogen so that organisms could use it, because ammonium, which was being converted into things organisms could use, was decreasing. The origin of biological nitrogen fixation and the timing of this origin is, however, debated. The antiquity of nitrogenase, the enzyme responsible for nitrogen fixation, points to an early origin, but it could have early on been used for something else, like, for example, the protection against hydrogen cyanide. Thankfully, however, regardless of when nitrogen fixation was able to come about, the ability for organisms to assimilate nitrate after methane and nitrogen from the atmosphere reacted to form hydrogen cyanide, which reacted in solution to form ammonium, which then was oxidized to nitrate by bacteria, allowed organisms to harness nitrogen in the Paleoproterozoic when nitrogen fixation was likely either not happening biologically yet and only by abiotic processes like lightning fixing nitrogen, or it had evolved, but it wasn't widespread yet. And because nitrogen is such an important nutrient, they needed to be able to assimilate this nitrate and use the nitrogen. Eventually, the accumulation of nitrate from nitrification, or the conversion of ammonium to nitrate, led to the invention of denitrification, which is exactly the opposite process, converting nitrate all the way back into molecular nitrogen, nitrogen 2, atmospheric nitrogen, went back into the atmosphere. And that was then used by the nitrogen fixers, who then completed the nitrogen cycle. So it became a nitrogen cycle early on Earth when these guys evolved these metabolisms. And as oxygen continued to increase, organisms could then harness it for higher energy yield, right? I mean, it makes sense if we look at the redox tower over here, basically showing that oxygen has the most reduction potential of all of these things on the redox tower, which makes it a very powerful compound to use in metabolism because it can oxidize all of these things above it, which allow organisms to gain energy from that process. However, organisms couldn't just pick up oxygen and use it because of its higher energy yield. They had to evolve the metabolism to use it. And not only that, they also had to evolve the required enzymes, because oxygen respiration involves some reactive intermediate oxygen species like superoxide and hydroxyl radicals that are really unhealthy for organisms, including ourselves, which is why we like things called antioxidants. We don't want these harmful intermediate products, harming us. And neither did the organisms that first started using oxygen respiration. So they had to evolve antioxidant enzymes, which now, we have and use in our bodies, and so does everything that uses oxygen respiration. However, as oxygen rose, these enzymes and this metabolism might have already evolved. There is evidence that oxygen respiration as a metabolism predates the evolution of cyanobacteria, which produced the oxygen that necessitated the oxygen metabolism, but this is still debated, and it wouldn't have been a lot of organisms that used that early on. It wasn't until cyanobacteria caused the oxidation events of Earth to make oxygen respiration really necessary and, widespread and diverse. But an increase in oxygen also triggered the diversification of chemolithoautotrophic pathways other than oxygen respiration. Because oxygen does have such a high reduction potential and ability to oxidize these compounds above it on this redox tower here, reduced compounds like methane, reduced sulfur compounds, iron 2, reduced iron, reduced manganese, ammonium, and nitrite could all now be usable in metabolisms because they could be oxidized by oxygen. So all of these organisms were able to oxidize these chemicals with oxygen to gain energy and live. And then eventually organisms evolved the ability to oxidize more complex organic carbon compounds like glucose, which is how we metabolize. We use oxygen and reduced organic carbon or glucose, and we oxidize that organic carbon with the oxygen, and then we release oxidized carbon or CO2. This became a cycle in which oxygen transform reduced compounds into food for reducers because it oxidized those compounds into reducible compounds like sulfate, oxidized or ferric iron, nitrate, and oxidized manganese, which oxygen did this in two ways, both abiotic, oxidative weathering and transport to the ocean, and biological oxidation, like we mentioned on the last slide, where all those compounds could be oxidized by oxygen in these biological organisms' metabolisms. And then the reducers, the biological reducing organisms, turned it back into food for the oxidizers, and it became a cycle. The oxygen was coming from primary producers, aka photosynthesizing cyanobacteria, that took light energy and converted it to chemical energy to reduce carbon dioxide into organic carbon that they could use to build their biomass, and then in the process split water to release oxygen, and that oxygen was then used by oxidizers to oxidize the reduced chemicals into oxidized chemicals for the reducers. And then reducers used hydrogen from hydrothermal vents, but could also eventually use organic carbon, which was on early Earth, abiotically formed occasionally, but only very simple organic carbon compounds were abiotically formed. And then later, they could use the organic carbon from other organisms as well. But early on, they also did autotrophic pathways by using hydrogen as their electron donor to reduce these compounds. And due to this complementary relationship that reducers had with oxidizers, they were able to stratify themselves into adjacent communities, microbial communities in microbial mat environments. And I have a full video on microbial mats if you want to check it out. I'll link to the top right for you. Eventually, however, supply of oxygen exceeded the supply of reductants. And we can see, I just increased the arrow size over here so you could see that oxygen just overwhelmed this cycle at some point. And at that point, all the chemical sinks or reduced compounds that were being oxidized by the oxygen that was increasing on Earth at the time were sufficiently oxidized. And obviously, like I said, the supply of oxygen overwhelmed that, and then boom, you had the GOE because of that. So that is the point at which oxygen rose very rapidly in concentration on Earth because it hit this boundary of, oh, now I've exceeded the supply of reduced compounds, and I've hit this wall where instead of oxidizing those compounds, I'm now going to build up as molecular oxygen instead of reacting with things. And that is when our atmosphere changed drastically and became more rich in molecular oxygen. And we know the timing of this GOE, or Great Oxidation Event, is around 2.4 to 2.1 billion years ago because of BIFs, or banded iron formations, the MIF signal of sulfur isotopes, which is a shift in sulfur isotopes that indicate the oxidation of the atmosphere, reduced beds before the event and red beds after the event, or basically detrital pyrite type things before the event, which couldn't have happened after oxidation of the atmosphere, and then red beds or oxidized beds after the event. Sulfate levels in the ocean increased due to oxidative weathering after the Great Oxidation Event. And a lot of other isotopic evidence, like later I'll show a slide with carbon isotope excursion, and a lot of other isotopic excursions point to this Great Oxidation Event and its timing at this point in Earth's history. And also, obviously, biological shifts. Because once this event happened, we had much different metabolisms before and after that dominated Earth. However, I don't want to go into detail about these pieces of evidence for the timing of the GOE here, because I have a whole video about the GOE where I go over this evidence in detail in that. So I'll link it up here to the top right. But what I want to talk about here is really the impact this had on the biosphere. So there was mass extinction due to the GOE, because before the GOE, the biosphere was dominated by anaerobes, many of which were not just anaerobes that didn't use oxygen, but also they were obligate anaerobes, meaning they just couldn't stand oxygen. It was toxic to them. And the sudden increase of oxygen during the GOE caused the extinction of many species, up to around 90%. It's hard to tell exactly because the fossil record isn't so complete during this time, and these organisms were not always that well preserved during this time, but the estimates are around 90%, which is really crazy for a mass extinction. That's just a lot of organisms to go extinct. But O2 was not only toxic to the anaerobic chemoautotrophs and chemoheterotrophs that we've been talking about, but also to the cyanobacteria themselves. I mean, if you think about it, you wouldn't really want to be sitting in an atmosphere of your own waste because oxygen is, like their waste product. So it makes sense, but this also really strained cyanobacteria themselves. And it wasn't until respiring organisms came about to balance the oxygen production of cyanobacteria with oxygen consumption by, animals like us that breathe in oxygen, that the cycle really started to become relatively stable. And that had a long time to do so. I mean, there was the Great Oxidation Event 2.4-ish billion years ago, then glaciation, snowball Earth events because of ramifications of climate change, because of oxidation and carbon burial. And then you had decline of organisms, not only because of oxygenation, but also because of snowball Earth events. And then finally, like 1.5 billion years later, you had another increase in oxygen, more snowball Earth events, and then finally, like a relative stabilization of oxygen and carbon cycles when animals evolved in the Cambrian 500 million years ago. Yeah, that's a long time from 2.4 billion years ago. And finally, things started to stabilize at that point. But anyway, there was a mass extinction of anaerobes at the time. However, there was another mass extinction. And this was totally unrelated to just oxygen being toxic. Instead, it had to do with kind of a feast to famine type of a trend. So what do I mean? Well, isotope anomalies such as the carbon isotope signatures here, as well as barium and sulfur isotopes, I believe also show this anomaly immediately after the GEO that shows a rapid reduction in primary productivity of up to 80% reduction of primary productivity, or basically the photosynthesizing cyanobacteria. And based on what we see, it was probably a result of a change in the availability of nutrients, most significantly phosphorus. And so because of a reduction of the availability of phosphorus, there was a huge drop in primary productivity indicated by the end of the Lamagundi carbon excursion here that showed a big drop in carbon isotopes because of a big drop in carbon burial rate due to the drop in primary productivity. And this is why the GEO marks a tale of feast to famine. Early photosynthesizers were not yet balanced out by oxygen respirers, like I mentioned earlier, and they feasted on carbon dioxide and their essential nutrients like phosphorus to the point they exhausted their nutrient supply, and it couldn't replenish itself fast enough to keep up with them. And this caused the extinction event, the second extinction event, other than the extinctions that were caused by the initial increase in oxygen to anaerobic bacteria that didn't like the oxygen. And instead of recovering from this extinction event, like other mass extinctions show immediately after the event, there was a long period where the amount of life on Earth remained very small and low in diversity relative to later periods. And this was not a short period. It was about 1 billion years, and it's actually nicknamed the boring billion. And so we're talking more right after the Great Oxidation Event, so potentially expanding this brown region to include this until, like 2.1-ish, 2.2-ish billion years ago, when the Great Oxidation Event had its little dip downward after the actual, spike in oxidation. And then that whole period was lack of recovery of organisms until boom, later in the Neoproterozoic, like 800 to 500 million years ago, you had an increase in life and biodiversity, and first animals appear, which balanced out the carbon and oxygen cycles, and it finally led to a world that is more similar to what we know today. There were other oxygen ramifications, however. Some were beneficial too. So let's talk about what else happened due to this oxygen increase. Well, the ozone layer formed and allowed phytoplankton to inhabit the euphotic zone of the ocean because they were no longer being bombarded by UV rays that damaged their DNA. Terrestrial environments also started to become inhabited by microorganisms in the form of biofilms, which started to colonize terrestrial surfaces because they no longer required shelter in the soil due to UV radiation. There's evidence for a terrestrial biosphere, at least by microorganisms, in the way of higher weathering rates by 2.2 to 2 billion years ago, just after the oxidation event and formation of ozone. And then, like I have already mentioned, that the increased sulfate flux allowed SRB, or sulfate-reducing bacteria, to continue to dominate bottom waters of the ocean, and that allowed them to maintain anoxic or euxinic, sulfidic and anoxic, bottom waters of the ocean for a long time before the NOE or Neoproterozoic oxidation event. And so it's kind of like the atmosphere became pretty well oxygenated, at least to a certain level, by the GOE, the Great Oxidation Event, to something billion years ago. And then you had the Boring Billion, which included relatively oxygenated atmospheres with relatively anoxic and sulfidic bottom waters of oceans until the Neoproterozoic oxidation event, 700 million years ago, where both the atmosphere and the ocean became more oxygenated. And another thing that oxygen caused was eukaryote diversification. The timing of eukaryote evolution actually agrees pretty well with the GOE. So around 2.1 billion years ago is the oldest eukaryote to our, findings. And then the NOE marks when eukaryotes began to really diversify and become more complex due to higher energy yields because of oxygen metabolisms. But like I just mentioned, the bottom waters of the ocean remained largely not oxic until much later after the GOE. And I mentioned BIFs earlier, banded iron formations, and this is one major reason we used to think that the GOE marked the full oxygenation of both the atmosphere and the oceans, but now we know that's just not true. The termination of banded iron formation deposition around 1.8 billion years ago after the GOE used to be attributed to fully oxic bottom waters because the iron would have been fully removed by oxygen. However, more evidence now points to the termination of BIF formation because of the removal of iron by sulfide instead of oxygen, which also agrees with the fact that SRB, sulfate-reducing bacteria, were maintaining the bottom waters really sulfidic during the time after the GOE due to increased sulfate flux to feed them their sulfate food. And they also were producing the sulfide and keeping it anoxic and sulfidic. And that removed the iron, not the oxygen. And yeah, bottom waters remained anoxic and depleted in oxygen long after BIFs terminated, which goes against what we had originally hypothesized, but now we know better. And like I mentioned a couple slides ago, eukaryotes began to not only evolve, but also diversify during and after these oxygenation events. And actually, along with the formation of the first supercontinent, Rodinia, oxygenation caused major glaciation. And these snowball Earth events, which there were few major snowball Earth events that I talk about in my snowball Earth events video that I'll link up here to the top right if you want to check out. They devastated life, but they might have mediated the evolution of multicellular life. After the snowball Earth events, or at least the second couple snowball Earth events that coincided with this first supercontinent and the Neoproterozoic oxidation, there was major diversification of eukaryotic life in the form of animals. First, we had the Neoproterozoic soft-bodied Ediacaran fauna, and then the more skeletonized, hard parts secreting Cambrian organisms that were larger, more complex, and just created a whole new ecosystem of Earth's seafloor in the Cambrian. And that is what I discussed in my Cambrian explosion video. And then also I talk about the mediation of multicellular life in my Snowball Earth video. If you want to check those videos out, they have a lot of really cool information in them as well. And I will link them up here on the screen where, their squares are right now for you to click, and they'll pop up any second now, as well as the GeoBio playlist will pop up any second now. But if you want to check out my major reference for this and other videos in my GeoBio playlist, I'm using the Introduction to Geomicrobiology by Kurt Konhauser. It's linked in my description. And for this video, I'm also using Earth System History, which is also linked in my description. And also, guys, if you want to join my channel for extra content as a channel member, you can do so now. And I'd greatly appreciate your support. But anyway, you can support me even just by liking this video or commenting is greatly appreciated. I love you guys. Anyway, thanks for watching, and I'll see you guys next time. Bye.